Hello dear students, welcome to lecture series in Botany. Today we are going to discuss an important topic, that is glycolysis. In this lecture we will discuss about glycolysis, different reactions that take place in this pathway and then its regulation. Glycolysis is derived from Greek word glykos meaning sugar and lysis meaning splitting. So glycolysis means the splitting of sugar. It is the process whereby glucose, the most abundant monosaccharide in living systems, is converted to simpler products. In the early steps of glycolysis, carbohydrates are converted to hexose phosphates, which are then split into two triose phosphates. In the subsequent energy conserving phase, the triose phosphate are oxidized and rearranged to yield two molecules of pyruvate. Besides preparing the substrate for oxidation in the citric acid cycle, glycolysis yields a small amount of chemical energy in the form of ATP and NADH. The glycolysis was the first metabolic pathway to be elucidated and best understood. The development of methods of enzyme purification, the discovery and recognition of the enzymes such as NAD, and the discovery of the metabolic role of ATP and other phosphorylated compounds all came out of the studies of glycolysis. Glycolysis is an universal central pathway of glucose catabolism. The glycolytic breakdown of glucose is a sole source of metabolic energy in some mammalian tissues and cell types such as erythrocytes, renal medulla, brain, and sperm. Some plant tissues that are modified to store starch and some aquatic plants derive most of the energy from glycolysis. Many anaerobic microorganisms are also entirely dependent on glycolysis. Glycolysis occurs in all living organisms, prokaryotes as well as eukaryotes. The principal reactions associated with the classic glycolytic and fermentive pathways in plants are almost identical with those of all animal cells. However, plant glycolysis has unique regulatory features as well as parallel partial glycolytic pathway in plastids and alternative enzymatic routes for several cytosolic steps. In animals, the substrate for glycolysis is glucose and the end product is pyruvate. Sucrose is the major translocated sugar in most plants and is therefore the form of carbon that most non-photosynthetic tissues import. Thus, sucrose and not glucose can be argued as the sugar substrate for plant respiration. In the early steps of glycolysis, sucrose is broken down into two monosaccharides, glucose and fructose, which can readily enter the glycolytic pathway. Two pathways for the degradation of sucrose are known in plants. In most plant tissues, sucrose synthase localized in the cytosol is used to degrade sucrose by combining sucrose with UDP, that is uridine diphosphate, to produce fructose and UDP glucose. UDP glucose pyrophosphorylase then converts UDP glucose and pyrophosphate to UTP, that's uridine triphosphate, and glucose 6-phosphate. In some tissues, invertases present in cell wall, vacuole, or cytosol hydrolyze sucrose to its two component hexoses, glucose and fructose. The hexoses are then phosphorylated in the reaction that use ATP. Plastids such as chloroplasts or amyloplasts can also supply substrates for glycolysis. Starch is synthesized and catabolized only in plastids, and carbon obtained from the starch degradation enters the glycolytic pathway in the cytosol, primarily as hexose phosphates or triose phosphates. Plastids convert starch into triose phosphates using a separate set of glycolytic isozymes that convert hexose phosphates to triose phosphates. The glycolytic pathway is often referred to as Embden-Meyerhof-Prennes pathway in the honor of three biochemical pioneers, Gustav Embden, Otto Meyerhof, and Jacob Prennes. Glycolysis consists of 10 enzyme-catalyzed reactions. The pathway can be broken down into two phases, preparatory phase and payoff phase. Now the first phase of glycolysis, that is preparatory phase. In the initial phase of glycolysis, each hexose unit is phosphorylated twice and then split into two molecules of triose phosphate. The series of reactions consumes 2 or 4 molecules of ATP per sucrose unit depending on whether the sucrose is split by sucrose synthase or invertase. In this phase, glucose and fructose are phosphorylated and cleaved into 2 molecules of glyceraldehyde 3-phosphate. These reactions also include 2 of the 3 essentially irreversible reactions of glycolytic pathway that are catalyzed by hexokinase and phosphofructokinase. This enzyme is one of the control points of glycolysis in both animals and plants. Now the first reaction, that is hexokinase reaction. The enzyme hexokinase is present in most cells. In liver, glycokinase is the main hexokinase which prefers glucose as substrate. In this reaction, phosphorylation of hexoses, that is glucose and fructose, but mainly glucose, occurs to glucose 6-phosphate. It requires magnesium ATP complex as substrate. Uncomplexed ATP is a potent competitive inhibitor of this enzyme. This enzyme catalyzes the reaction by proximity effect, bringing the two substrates in close proximity. The enzyme undergoes large conformational change upon binding with glucose. This enzyme is inhibited allosterically by glucose 6-phosphate. Now the second reaction, phosphohexose isomerase reaction. The enzyme phosphohexose isomerase catalyzes the reversible isomerization of glucose 6-phosphate to fructose 6-phosphate. This enzyme requires magnesium divalent positive ions for its activity. It is specific for glucose 6-phosphate and fructose 6-phosphate. Now the third reaction, phosphofructokinase 1 reaction. The enzyme phosphofructokinase 1 transfers the phosphoryl group from ATP to carbon number 1 of fructose 6-phosphate to produce fructose 1,6-bisphosphate. This step is an important irreversible regulatory step. The enzyme phosphofructokinase 1 is one of the most complex regulatory enzymes with various allosteric inhibitors and activators. ATP is an allosteric inhibitor and fructose 2,6-bisphosphate is an activator of this enzyme. ADP and AMP also activate phosphofructokinase 1, whereas citrate is an inhibitor. Now fourth reaction, that is aldolase reaction. The enzyme aldolase catalyzes the cleavage of fructose 1,6-bisphosphate into glyceraldehyde 3-phosphate and dihydroxyacetone 3-phosphate by aldol condensation mechanism. The standard free energy change is positive in the forward direction. Now the fifth reaction, triose phosphate isomerase reaction. The enzyme triose phosphate isomerase causes interconversion of dihydroxyacetone 3-phosphate to glyceraldehyde 3-phosphate. This is a reversible reaction catalyzed by acid-base catalysis. Now the second phase of glycolytic pathway, that is payoff phase. Once glyceraldehyde 3-phosphate is formed, the glycolytic pathway can begin to extract usable energy in the energy-conserving phase. The enzyme glyceraldehyde 3-phosphate dehydrogenase catalyzes the oxidation of the aldehyde to a carboxylic acid, reducing NAD positive to NADH. This reaction releases sufficient energy to allow the phosphorylation of glyceraldehyde 3-phosphate to produce 1,3-bisphosphoglycerate. The phosphorylated carboxylic acid on carbon number 1 of 1,3-bisphosphoglycerate has a large standard free energy of hydrolysis. Thus, 1,3-bisphosphoglycerate is a strong donor of phosphate group. In the next step of glycolysis, catalyzed by phosphoglycerate kinase, the phosphate on carbon number 1 is transferred to a molecule of ADP, yielding ATP and 3-phosphoglycerate. For each sucrose entering the pathway, 4 ATP molecules are generated by this reaction. This type of ATP synthesis is traditionally referred to as substrate level phosphorylation, and it involves the direct transfer of phosphate group from the substrate to the ADP to form ATP. The ATP synthesis by substrate level phosphorylation is distinct from ATP synthesis by ATP synthesis involved in the oxidative phosphorylation in mitochondria and photophosphorylation in chloroplast. In the following reaction, the phosphate on 3-phosphoglycerate is transferred to carbon number 2 and a molecule of water is removed, yielding the compound phosphoenol pyruvate. The phosphate group on phosphoenol pyruvate has a high standard free energy of hydrolysis, which makes phosphoenol pyruvate an extremely good phosphate donor for ATP formation. Using phosphoenol pyruvate as substrate, the enzyme pyruvate kinase catalyzes a second substrate level phosphorylation to yield ATP and pyruvate. This phase includes 5 reactions. The first reaction of this phase, glyceraldehyde 3-phosphate dehydrogenase reaction. The enzyme glyceraldehyde 3-phosphate dehydrogenase causes conversion of glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. This is the first reaction of this energy yielding phase. Oxidation of aldehyde derives the formation of high energy acyl phosphate derivative. An inorganic phosphate is incorporated in this reaction without any expense of ATP. NAD positive is the cofactor in this reaction, which acts as an oxidizing agent. The free energy released in this oxidation reaction is used in the formation of acyl phosphate. Now another reaction, phosphoglycerate kinase reaction. The enzyme phosphoglycerate kinase causes transfer of phosphoryl group from 1,3-bisphosphoglycerate to ADP, generating ATP and 3-phosphoglycerate. The name of this enzyme indicates its function for reverse reaction. It catalyzes the formation by proximity effect. This step generates ATP by substrate level phosphorylation. Now the next reaction, phosphoglycerate mutase reaction. The enzyme phosphoglycerate mutase catalyzes the conversion of 3-phosphoglycerate to 2-phosphoglycerate. There is a transfer of phosphoryl group from enzyme to 3-phosphoglycerate, generating enzyme-bound 2,3-bisphosphoglycerate intermediate. This compound has been observed occasionally in the reaction mixture. In the last step of reaction, the phosphoryl group from carbon number 3 of the intermediate is transferred to the enzyme and 2-phosphoglycerate is released. Now the next reaction, enolase reaction. The enzyme enolase causes dehydration of 2-phosphoglycerate to phosphoenol pyruvate. Dehydration of 2-phosphoglycerate by this reaction increases the standard free energy change of hydrolysis of phosphoanhydride bond. Now the next reaction, that is pyruvate kinase reaction. The enzyme pyruvate kinase catalyzes a transfer of phosphoryl group from phosphoenol pyruvate to ADP, generating ATP and pyruvate. This is the second substrate level phosphorylation reaction of glycolytic pathway. This enzyme couples the free energy of phosphoenol pyruvate hydrolysis to the synthesis of ATP. The enzyme requires magnesium divalent positive ions and K positive ion. Now energetics of glycolysis. In the overall glycolytic process, one molecule of glucose is converted to 2 molecules of pyruvate. 2 molecules of ADP and PI are converted to 2 molecules of ATP. 4 electrons as 2 hydride ions are transferred from 2 molecules of glyceraldehyde 3-phosphate to 2 NAD positive to form NADH. 2 molecules of ATP generated can directly be used for doing work. The 2 NADH molecules are oxidized in mitochondria under aerobic conditions, and the free energy released is enough to synthesize 6 molecules of ATP by oxidative phosphorylation. Under the aerobic condition, pyruvate is catabolized further in mitochondria through pyruvate dehydrogenase and citric acid cycle, where all the carbon atoms are oxidized to carbon dioxide. The free energy released is used in the synthesis of ATP, NADH and FADH2. Under anaerobic conditions, pyruvate is converted to lactate in homolactic fermentation or ethanol in alcoholic fermentation. Now the regulation of glycolysis. 2 types of controls are present for metabolic reactions: substrate limited, enzyme limited. Now the first, substrate limited. When concentration of reactants and products in the cell are near equilibrium, then it's the availability of substrate which decides the rate of reaction. Now enzyme limited. When concentration of substrate and products are far away from the equilibrium, then it's the activity of enzyme that decides the rate of reaction. These reactions are the one which control the flux of the overall glycolytic pathway. There are 3 steps in glycolysis that have enzymes which regulate the flux of glycolysis: the hexokinase, phosphofructokinase and pyruvate kinase. Now the first one, hexokinase. Hexokinase performs first step of glycolysis in most tissues including muscle and brain. It has a high affinity for glucose, so it permits initiation of glycolysis even when blood sugar levels are relatively low. Hexokinase is inhibited by the product of the reaction, that is glucose 6-phosphate. This is a very important regulatory step since it prevents the consumption of too much cellular ATP to form glucose 6-phosphate when glucose is not limiting. Glucokinase found in liver and pancreatic B cells requires a much higher level of glucose for maximal activity. It is thus most active when glucose is very high in the portal vein immediately after consumption of a carbohydrate-rich meal. Glucokinase is not inhibited by glucose 6-phosphate. The action of glucokinase is opposed by the action of glucose 6-phosphatase, which hydrolytically removes phosphate in the futile cycle. At normal levels of glucose, The forward and backward reactions are balanced. Glucokinase activity is also affected by an inhibitory binding protein, whose affinity for glucokinase is enhanced by binding fructose 6-phosphate and lessened by binding fructose 1-phosphate. Fructose consumed in the diet is converted to fructose 1-phosphate in the liver. This opposes the action of fructose 6-phosphate on glucokinase, favoring the forward reaction. The capacity of liver cells to phosphorylate fructose exceeds their capacity to metabolize fructose 1-phosphate. Consumption of excess fructose can cause an imbalance in liver metabolism by indirectly depleting liver cells of ATP. Now the second one, phosphofructokinase 1. Phosphofructokinase catalyzes the rate-limiting step in glycolysis and is the most important control point. It is also the first irreversible step that is unique to the glycolytic pathway. Glucose 6-phosphate can be used as an intermediate in other pathways including glycogen synthesis and the pentose phosphate pathway. Phosphofructokinase is allosterically inhibited by ATP. So, glycolysis is slowed when cellular ATP concentrations are high. ATP binds to the site on phosphofructokinase, distinct from the active site, causing a conformational change. The conformational transition is regulated by cellular pH. Glycolysis is also a means of preparing carbon-containing backbones for biosynthetic reactions in the form of acyl coenzyme A. Citrate is an intermediate in the TCA cycle, also inhibits phosphofructokinase. Phosphofructokinase is allosterically activated by high levels of AMP, that is adenosine monophosphate. AMP also overcomes the inhibitory effect of ATP. Another allosteric activator of phosphofructokinase is fructose 2,6-bisphosphate. Fructose 2,6-bisphosphate is not an intermediate in the glycolytic pathway. It overcomes the inhibitory effect of ATP. Fructose 2,6-bisphosphate is made from fructose 6-phosphate by a specific kinase, that is phosphofructokinase 2. The same polypeptide that encodes this kinase also encodes a phosphatase that catalyzes the reverse reaction. Activity of this bifunctional enzyme is controlled by phosphorylation. Phosphorylation of the bifunctional enzyme is regulated by blood glucose level mediated by glucagon and insulin. High glucagon, that is low blood sugar, causes phosphorylation of the enzyme, which results in the conversion of fructose 2,6-bisphosphate back to fructose 6-phosphate, removing its stimulatory effect on phosphofructokinase and therefore slowing the rate of glycolysis. Conversion of fructose 6-phosphate to fructose 2,6-bisphosphate is also stimulated by high levels of fructose 6-phosphate. This is an example of feed-forward stimulation. Fructose 2,6-bisphosphate is also an important regulator of the process of gluconeogenesis, where glucose is synthesized from pyruvate. Now third one, pyruvate kinase. Pyruvate kinase is the third regulated enzyme of glycolysis. Like phosphofructokinase, pyruvate kinase is regulated both by allosteric effectors and by covalent modification. Pyruvate kinase is activated by fructose 1,6-bisphosphate in the liver, A second example of feed-forward stimulation. ATP and alanine act as allosteric inhibitors of pyruvate kinase. Phosphorylation of pyruvate kinase is regulated by blood glucose level, just like phosphofructokinase. High glucagon causes phosphorylation, which in this case renders the enzyme inactive. Pyruvate kinase deficiency causes hemolytic anemia, since red blood cells rely entirely on glycolysis for ATP synthesis. In this inherited disorder, pyruvate and lactate levels are lower than normal, while proximal intermediates such as 1,3-bisphosphoglycerate and phosphoenol pyruvate accumulate. Hormonal regulation of glycolysis ensures coordination among different tissues and organs. So friends, with this we conclude today's program, and I hope you might have understood this topic. See you next time with another topic. Till then, goodbye.